hope you're enjoying the ATTIS. And I'm up next. We are a few minutes behind schedule, but I think we'll end up catching up, and everybody will get the amount of time that they were allotted. So now we are ready to start the next phase of our program, which is our flash talks from our trainees. So I will share my screen and show that. Go. Okay, so each person will have 10 minutes to talk, and you don't have to take up the whole time. And I will give you warnings when you have when you're halfway, and 2-minute, 1-minute warnings. And so then we'll want to stop to give everybody enough time to talk. So our first talk, the title actually changed. So the title here may not be the title that you have in your program, but the title was updated, and it's Ari Ginsparg, and I apologize in advance for mispronouncing anybody's name. The name of the talk is New Algorithms to Discuss Chemical Modulators of Sleep Drive.
Well, all right. Is my presentation up?
Yes.
Okay, perfect. Yeah. Yeah. So hi, I'm Ari Ginsparg. I'm in Dr. Summer Times Lab here at UAB in the Graduate Biomedical Sciences program. So here I'll go over a overview of what my thesis project is. Yeah. So here's a overview, a schematic of everything that I'm working on. So essentially, what I'm doing is working in the field of drug discovery. I've developed a algorithm in the Rosetta protein modeling suite that looks at a target protein, and the algorithm tries to play matchmaker with a library of ligands, and then with that library with the algorithm, it outputs predictions of how the ligands fit in the binding pocket. And so with that, the time lab specializes in using zebrafish as a model to study neurological diseases. And so with that, with outputs of predicted placements from the algorithm, I want to look at drugs that look to be of interest and test how they work in a zebrafish. So like a example that I'm looking at is sleep in a zebrafish, and specifically looking at remedying narcolepsy. So currently we're in a drug discovery revolution. There's been a lot of recent pursuits in drug discovery, and it's thanks in part to two things. One is the development of massive libraries, such as the real enamine library, as well as the development of ligand docking software, such as the program that I've developed, or other popular softwares like Autodoc Vina. And so the library that I mostly focus on right now is the real enamine library, which currently has a catalog of over 2 billion synthesizable ligands, and this library has already proven to be the source of molecules that target various proteins, including GPCRs, which is what I'm interested in. So as for the algorithm that I've developed, so I developed it in the Rosetta protein modeling suite. And so what Rosetta is that it's a large collaborative effort. There's over like 500 developers working on it. And so there's a few hallmarks as to why I've decided to choose Rosetta to develop in. First is a state-of-the-art energy function, which is really good for evaluating the stability of a protein ligand system. Next is their highly modular code architecture. They have a bunch of classes and data structures that represent like proteins and ligands, as well as do things like measure the energy of the system. And the biggest thing that's unique to the algorithm that I developed is that it uses a data structure called a motif, which is a representation of 6 atoms, 3 from a amino acid and 3 from a ligand. and so with that, I derived a library of 1 million motifs from existing protein ligand systems from the protein data bank. Uh, and, and so with that, I'll demonstrate how this this motif method works. So here we have a example protein ligand system from the PDB, and so the bolded atoms are the atoms that compose the motif. So it's the carbon and 2 nitrogens here, and then the phosphorus and 2 oxygens here. And so then we have our target PDB, and so my algorithm focuses on a user specified residue in the binding pocket that we then use as a anchor. And so basically, we go through all motifs that work for a given residue, and then we also iterate through the inputted library of ligands. And so basically, the matchmaker part that happens is that we align the atoms in the ligand to the ligand atoms in the motif, and then from there we get a place ligand that we then evaluate for in clashing it against the backbone. And so with that, so I've currently done some benchmarking as well as comparisons of my algorithm against a popular docking algorithm, AutoDoc Vina. And so the comparison that I did was that I use 91 existing protein ligand systems to compare the ability for both of these algorithms to predict the native placement of the ligand. And so I took a set of the 100 best placements based off of a free energy score, and then collected the RMSD of these predicted placements to what the native placement looks like. And so with this comparison, the motifs method that I use that uses the information on native systems, it appears to outperform AutoDoc Vina. And so with that, the main metric that I use, I was taking account of a number of placements that had a RMSD that was compared to the native placement that was less than 2 angstroms. From looking at this, or from looking at the placements, a RMSD that's 2 angstroms or smaller, it generally conserve the intermolecular interactions between the protein and ligand. And so with that, the motifs algorithm had a 85% rate of placing within 2 angstroms, whereas AutoDoc had a 64% rate of placing within 2 angstroms. And so and that's seen here and here. And then here we have a scatter plot that plots the 91 different systems with the best RMSD per system. And so like points down here in this corner show that our systems where both algorithms did well. Systems here are ones that the motifs method did well, and then systems here are ones that the AutoDoc did well. And so
you have just under 3 minutes left.
Okay, perfect. I'm just about at the end. So as for a further direction to go from here with actually testing my algorithm. So as I said earlier, I'm specifically interested in sleep in a zebrafish, and so with that, my next big test case for the algorithm is this hypercretin receptor, which is found in vertebrates and is fairly well conserved between humans and zebrafish. And with that, the underactivation of this receptor leads to narcolepsy. And currently, there hasn't been a agonist drug that's been discovered to activate this receptor. And so what I'm going to do is run the adenine library through my algorithm and try to come up with a potential agonist, which I will then test in a zebrafish and monitor their sleep behavior. And then finally, if we do get a drug that looks like it remedies a narcolepsy phenotype, then I'll try to derive a cryo-EM structure of the ligand-bound hypercretin receptor, and that'll help to validate the prediction accuracy of my algorithm. And so that's pretty much it. I have a lot of people to thank for helping me along the way with this project. Yeah, and so I'm not sure if there's like questions at this point, but that's basically it for my presentation. Thank you for listening.
Any questions? Feel free to unmute yourself, or you can enter them in the chat. We have about a minute left.
Cool.
What are some of the kind of the major challenges that you ran into?
I like the biggest challenges with coming up with the algorithm is like I had to familiarize myself with the whole Rosetta code base, and then from there, like it definitely was some issues. I was just kind of like tuning the algorithm and make sure that it actually works with placing the ligands, and then yeah, and then also like being able to calculate the energy scores of the systems.
Okay, thanks. Great presentation. Thanks.
Thank you.
Let me introduce the next speaker. The next speaker is Louis Paulin, and the name of the talk is Comprehensive Structural Variant Detection from Population to Mosaic Level. I can see your screen. You may be on mute.
Oh, sorry. Yes. So I'm Luis Paulin.
Perfect.
I'm a postdoc at Fritz Selacek Lab at Baylor College of Medicine. And today I will talk to you about SNIFL2 and how it enables comprehensive structural variation detection from mosaic to population level. So structural variants are roughly defined as 50 base pairs or larger alterations that come into 5 types: deletions, insertions, duplications, inversions, and translocations. And in many cases, we have kind of like combinations of multiple of these. And I'll show you some examples later. And even though single nuclear variation are in the roughly like 4 to 5 million per human genome, structural variants are kind of the largest amount of variation in the human genome, given their size. And they impact all parts of evolution and biology. So in evolution, they have caused speciation events. They also important genomic disorders where they have been linked to Mendelian disorders, neurological disorders, and cancer. Also, they impact gene regulation because there can be duplications that will alter gene dosage, and thus will affect how the cells behave. And finally, they also have an impact on phenotypes. Here, if we add the structural variations to the overall variation, we can better explain many traits. Here are some examples that explain heritability. And in red, you will see that we have added a single nucleotide variation, but in gray and black is when we also add structural variation into the mix. So up to now, or like a few years ago, short reads was kind of the way to do all kind of analysis. However, when we compare like short read sequencing and structural variation, usually for human genome, we get from 10 to 12,000 structural variants per human genome. However, now with long read technologies, we're able to not only get more of those, almost twice of them, but also we can access difficult genomic regions that were not previously able to be detected. And these contain medically relevant genes, because they are kind of in highly repetitive sequences. And now we have access to the centromere and telomere. So in that regard, we have implemented here Sniffles2, which is kind of an upgrade of the previous and well-known structure variant detection tool Sniffles. And we have increased the accuracy of Sniffles2 when we compare to Sniffles1, and also we have made them faster. And not only that, but we have enabled new applications, such as in germline detection of large insertions and deletions. We also have implemented a population mode that we can provide a fully genotype BCF file for a family or a population in seconds. And finally, we have implemented a non-germline mode in which we can identify somatic or mosaic mutations. I will show you examples of the 3 of them. So for the first part of the benchmarks, what we make use of is the genome metabolic benchmarks. The first one is a genome-wide benchmark derived from sequence resolved datasets. And the second one is a challenging medical genes, which characterization of 273 genes that are not accessible with short read data. And here we have 200 structural variants for our test. And many of these genes are too complex because have a lot of repetitive sequences or have segmental duplications. So for the first part, for benchmarking Sniffles2, we can see here on the top Sniffles2. And what we have here measured is the genotype F1. So the F1, it combines accuracy and precision in a single measure. But here we're not only testing if the structural variant is present or not, but also that the genotype is correct. And we compare Sniffles2 to the Sniffles1 and other popular tools such as QSV, SVM, and PBSV. And what I can show you here is that in blue, Sniffles2 has the best F1 genotype score and also the lowest CPU time. So here's in minutes. We tested it across several coverages from 5 to 50x, and you can see if with 5x, many of these tools cannot even give an answer. And on the other hand, when we increase coverage quite high, PBSV, for example, in purple, did not finish the last round. So it's not plotted here. So here we have shown the Sniffles2 perform fast and is the most accurate of all. But here we have been using default parameters for all tools. So in order to give them a chance to compete with our kind of new tools, Sniffles2, we optimize parameters for all colors. In some cases, for each of these structural variant colors, you need to specify some parameters for coverage and even for technology. So Sniffles2 has automated kind of parameter detection, and that's why it outperforms others at medium and low coverages. So when we optimize parameters, we can see that still in many cases now they're able to compete, but never achieve the same performance as Sniffles2. Next, we went to the challenging medical genes. As if you can see here, the F1 genotype score is way lower because these regions are very challenging. However, Sniffles2 is still the most accurate and the fastest of them all. So when we compare results of other tools to Sniffles2, here you can we show you the improvement of Sniffles2 to other tools, and here how much time other tools took to have the same result. So when we took the second most accurate tool here, QSV, for this result, it took roughly 10 times the time. And if in the other hand, we took SVM, so the second fastest one, we can see that Sniffles2 has like a roughly 12% higher accuracy in the score. So here we have shown that Sniffles2 has not only fastest and more accurate at other tools in 2 of kind of the top of the line benchmarks that are used to the moment. So with that aside, we prepare new modes for Sniffles2. The first one is a population mode, because in our recent review by our group, we show that the number of genomes that have been sequenced with long read data is exploding. As of January 2021, there were already in the thousands, and there are plans of using Oxford Nanopore technology to sequence 1 million genomes. So for that, we need a way to make a population BCF file fully genotype fast. Current methods are slow. They require several steps. For example, here, first you need the ESV calling of all the individuals you're gonna use for your population BCF file. Then we'll perform a first merge, in this case using a software called Survivor. But this is not the fully genotype BCF file. There will be many place as many structural variants, and we will not have a genotype. So there's a regenotyping tool in which we use the previous information of the merging and the information of the alignment to regenotype all these structural variants. And then we'll need to perform a second merge in order to have this population BCF file. We tested this with a family trio, and it took for this pipeline 36 hours, not counting the initial SV calling, just the merging, regenotyping, and merging again. So
you have a little over 2 minutes left.
Okay, so with Sniffles 2, we have performed like a new way to do it. And in this case, we merge. We have a specially binary file, which is like a GBCF file. And in a single step, we have a genotype, a full genotype. So for the same trio, it took roughly 65 seconds. And also we have solved the N+1 problem, which means that we can add samples as soon as we have it later when they are sequenced, and we can scale up to thousands of genomes. So let me show you first some of the results here for speed. So we showed that Sniffles 2 can combine samples in almost linear time, and we performed a stress test with 1,002 genomes, and it took 9.25 CPU hours, which is 4 times faster than it took skewed SV to perform a single family trio. So with this, what we did is we studied a population of 31 individuals from that contain 7 full families. And with this population merge, we were able to detect disease-relevant structural variants that affect the MECP2 region. And this is one of the examples.
I have 1 minute left.
We were able to detect here, for example, here we show you 2 inversions within a very complicated region. I will go to the end now, because this is another important thing that we perform. So what we also were able to do is to detect what we call somatic or non-germinal SVs, which are structural variants that are just in a couple of reads. And here you can see here 3 reads that are affecting a patient. And in this case, we were able with this non-germinal mode to detect these 3 structural variants without losing much precision in this case. And these non-germline are important in, for example, here, rare neurodegenerative diseases, so that were not able to be captured by the Illumina and DX or either by BioNano. So with this, go to the summary in which we have improved Sniffles for the detection of germline SVs, but we also have implemented other applications such as the population mode and the somatic SVs that are important, for example, here in neurodegenerative disorders. But software is already available in Conda, and the preprint is available at bioRxiv. And I would like to thank you for your attention. And any questions for the 30 seconds we have?
Unfortunately, we're out of time, so maybe you can, maybe we can chat. We can chat your questions.
Sure.
So sorry about that, but great talk.
No problem.
The next speaker is, is Christopher Grochowski, Baylor College of Medicine, complex inverted genomic triplication structures, 2 breakpoint junctions, several possibilities.
Okay, can you see my screen okay?
Yep.
Okay, so thank you so much for the opportunity to speak here. Again, my name is Christopher Grochowski. I'm a postdoctoral associate in the Department of Molecular and Human Genetics in the laboratory of James Lepsky here at Baylor College of Medicine. And I just wanted to talk about a little bit of our work that kind of started as my thesis work and is now continuing into my postdoctoral studies, titled Complex Inverted Triplication Structures, 2 Breakpoint Junctions, Several Possibilities. And so since this is a flash talk, I'm going to throw a lot of data really quickly, but I hope we can kind of catch up. So as Luis kind of showed in the previous presentation, structural variation encompasses very large changes to the genome. That can be duplications, deletions, inversions, translocations, but really what I want to focus on and what I focused on in my thesis work are inversions. So kind of starting at the top here, right, we can have inversions that present with 2 breakpoint junctions and a 180-degree flip of DNA. So this can be in a copy number neutral state or present as more complex SVs with copy number variation that's actually carried with the inversion itself. So there are actually more complex presentations that involve inversions, and one that we've studied very extensively in the laboratory is the duplication, triplication, inverted duplication structure. Here shown by an array CGH. And this structure was really first characterized by Claudia Carvalho in 2011 that showed that basically the triplication is in an inverted status between the duplications, forming two breakpoint junctions between the two structures. And this was what she found was that the structure was actually mediated by a given pair of inverted low copy repeats that was downstream of the structures. You can see here in the array CGH that's highlighted here, these yellow arrows are effectively pointing to the inverted low copy repeats that actually mediate this event. So when we look at kind of rare disease cohorts, we can actually find that this structure is very prevalent in many cohorts, including MECP2 duplication syndrome, Taki-Lepsky syndrome, and others. And they're all really stimulated by those inverted low copy repeats that causes the event to occur. And specifically, we've really focused on MECP2 duplication syndrome in the laboratory, not only here in the Lepsky lab, but also the Carvalho lab at the PNRI in Seattle. And what we found in our large biobank of samples is that about 17% of these samples actually have a dupe trip inverted dupe structure that basically spans the dosage sensitive gene MECP2. And what I think is even more fascinating is that when we look in the cancer genomics field, we can actually see this dupe trip inverted dupe structure is actually one of the 12 most prevalent mutational signatures that's also found in cancer genomes. So what we want to do is kind of really resolve this structure and understand the breakpoint junctions and understand the context of each genomic fragment in terms of the larger structure. The problem is that we are sitting with these low copy repeats. They're very difficult to see through, not only on an array CGH, but also different whole genome sequencing technologies. And we can kind of appreciate here, we have a large gap where we have basically a gene repeat, and we have gaps where our breakpoint junctions are occurring for the structure. And so what we really wanted to use is a multi-omics approach to try to understand the haplotype structures for these samples and understand the breakpoint junctions and exactly where they're occurring. And so what we did was basically apply as many technologies as we can to give us a different view of these structures, including array CGH, short read whole genome sequencing, long read sequencing, including nanopore and PacBio HiFi, as well as optical genome mapping, to really lean on the strengths of each of these technologies to try to piece together the structure for these samples. And kind of what I wanted to highlight over here on the left is what's really nice with something like optical genome mapping is that we have up to 700-800 kb lengths of intact DNA that gives us resolution to understand the context of each structure within this larger dupe trip inverted dupe. And so what happens is with the optical mapping is that we don't actually have nucleotide level resolution, but we have sequence motifs that we can then map together to understand which piece is inverted and connected to which other piece. And what happens is that we can build that with our short read whole genome sequencing, with our HiFi, with our Oxford Nanopore, to kind of gain a more complete picture of the aberration in its totality. And what's nice is that we can actually revise our structural variant haplotype to better classify each of these dupe trip inverted dupe samples and kind of stratify them down to more refined sub-haplotype structures. And so really astonishingly, when we start to look at these dupe trip inverted dupe structures, we can see that it's not just one classification, but there are actually 4 distinct and previously unknown subtype structures that we could identify. And so
you have 5 minutes.
Okay. What we started to do is that we started to go through and create prediction models, right? If we can understand exactly so here I kind of highlighted in green and purple, exactly what inverted low copy repeat was used as a recombinant substrate that led to the event. And what we can do is kind of follow this through to understand if we basically have a collapse fork here, we recombine with an inverted low copy repeat, and then we can generate this structure. And so we basically can create prediction models for a collapse at each of the inverted low copy repeats to understand these subtype structures and to really understand how each of them come together in the larger haplotype. And so what I kind of wanted to show here is, we have this region on XQ, and we have these MECP2, which is our dosage sensitive gene. We have our low copy repeats here, and we can really try to understand the context of what we see in our optical mapping and what we see in our hi-fi data to give us a better understanding of this structure and try to really refine the haplotype structure itself. And what I think this has relevance in is the study of Mendelian disease and cancer mutagenesis. And again, I showed that it's actually one of the 12 most prevalent cancer genome signatures. This data really confirms our hypothesis that inverted low copy repeats act as a recombinant substrate and really play a critical role in the formation of these dupe trip inverted dupe events. And so with that, I just want to thank the entire team, especially Claudia Carvalho and James Lupsky, who have been really phenomenal mentors throughout my thesis work and now my postdoctoral work, as well as Fritz and Luis, who we've really worked together. And there's a beautiful paper that was just submitted to bioRxiv that highlights kind of sniffles 2 that Luis was talking about earlier, that kind of helps to bioinformatically look at these very complex chromosomal rearrangements and try to piece them together in a way that has been very difficult previously. So thank you so much for the opportunity.
Yeah, thanks. We have about 3 minutes for questions. If anybody has any questions, don't be shy. You can chat, or you can just unmute yourself. Anybody have any questions? I know a lot of people are interested in this topic. Okay, things that Jen.
Yeah, thanks for coming, Chris. This is wonderful. It's great. So this, I didn't catch the point. So it's still targeted sequencing, right? For that region, or okay.
Well, for the hi-fi, right, we're not doing target. We did some targeted sequencing with nanopore, but for the hi-fi that we're doing, for the nanopore that we're doing mostly, it's not targeted for the region.
So it's a whole genome.
Correct.
Okay, wonderful. Thank you.
Yeah, we've gotten really, really nice data coming out, because again, those low copy repeats, you just get no information from short read. And so we kind of wanted to move to hi-fi, to nanopore, so that we have resolution for those low copy repeats that'll try to give us more information of where the breakpoint's occurring and what low copy repeat it's actually starting on.
Wonderful, thank you. I think to save time, there's some question in the chat. Just speak out, unmute yourself, probably. Curtis, you have some questions?
Yes, let me raise my hand.
Juan also have a question.
I guess I was asking about, this is Curtis, going from the candidate to the final assemblies. Was that a hand process, or is that something you guys managed to actually make happen?
So, I mean, I think the ultimate goal in the end is like a fully automated process that can kind of really go through. Right now, it's kind of a combination of both, right? Even working with Fritz's group and Sniffles too, right? They give us calls that are useful, but doesn't give context of the full structure in and of itself. So I think right now, it's kind of a combination of automated and by hand, but the kind of the final product of understanding the full structure is still a very manual process that I hope eventually will become more automated, because it is a very time-consuming process per sample.
And I was just saying, from a very naive standpoint, you know, given the structural differences between prokaryotic and eukaryotic genomes, what would be applicability of some of the tools that you and Luis have talked about for prokaryotic genome structural variation detection?
I guess I haven't thought of that question specifically, but I mean, I think presumably as long as the structural variation types and classes are similar, then it would be directly applicable and be able to be used across different genome types. And even these tools are being used on plant genomes, are being used for certainly outside of human. So I think it does have broad applicability outside of just human disease.
Okay, thanks. Unfortunately, we have to move on. So any questions, please put them in the chat. And so the next speaker is Radomir Slaminsky, bioinformatics graduate, associate in the School of Biomedical Science at Informatics Institute, UAB. And the name of his talk is A Case Study of Functional Genomics Downstream Analysis with the Pager Web App.
Am I on? Oh, okay. Share the screen. Can everyone see the screen?
Yep, and I can hear you too.
Sure, good. So, hi, my name is Reimer Saminsky. I'm a first year graduate student, Dr. Chen's lab, and what I'm presenting is a case study of functional genomic analysis with Pager web app. So a little bit of background GMPA. So there are many studies have shown that like in that early detection is critical for like treatment of diseases. And this is why like having a gene set network and pathway analysis is being developed, and it's a powerful tool. And there are several GMPAs that have already been developed, like David, EnrichR, WebGestalt, and of course Pager. And the one I'm focusing on is Pager. So Page, what it is a pathway annotate gene lists and gene signatures, and Pager is the pathway annotate gene list, but it's electronically signatured electric repository. And then finally Page web app, which takes it to another level. And we'll be discussing these three. So first we start with Page. So this is kind of like a super gene set, is a new data set to represent the like page. So each page is a set of genes in curation. So it's like, for example, pathways from RNA, seq, microarray, and proteomics, and then annotate gene lists. So it's like a list from RNA, seq, microarrays, and proteomics, and then finally gene signature. So it's from different sources. So it's a combined there. And what Pager is like electronic repository form. So what it shows here is like, this is like a Pager website here, how it looks, and then you just enter the gene, and then it takes the COCO score, and then it takes like the network. So it shows the different networks, and here, and so this is like, and finally the web apps. This is an interactive online application to perform gene sets, and this is featured like using underlying technology like Shiny. You'll call it network visualization, and so 4 steps in the Pager web app. First of all, is to load either the demo like data set, which we have under, or the data set of interest you have in text format with the symbol for gene name, log2, log change, and p-value. And then the second step is like a volcano plot using the gene log2 changes that will highlight the gene significance with the p-value, usually less than 0.5 and absolute log2 value greater than 1. And then both values can be adjusted according to needs. And then the third step is a gene-rich analysis that will be done. Usually default is p-type page. And then fourth is a Venn diagram that compares Pager, EnrichR, and WebGestalt. And here this just shows the diagram that, so you load the data and then you filter it using volcano plot, the enriching, and then finally to compare. And so now I'm gonna get into a case study, which is a comparison of the gene expression in healthy control versus multiple sclerosis. So for demonstrating it, we used like a study that uses 12 MS patients and 15 healthy control. And the first step is the volcano step process. So this shows like how it looks overall after we enter the data. So this is the red is a up, the blue is a up regular, and then there's the shows a different filtration of the data sets. And then for this, and then you go to the next step, it shows the top pathways. And the one I found according to this is the VEGF pathway, which is an angiogenesis pathway. Then there's also circadian rhythm and the miRNA targeted genes and lymphocytes. So it has to do with immune system function, B cell receptor. So it just shows you the networks in which can play a role in the MS. And then EnrichR related to differently expressed genes. This is based on the different databases like Reactome and then WikiPath and then KEGG. And then here it shows the different pathways that they discover like VEGF, circadian rhythm, and then MIR. And then the PAG network is, which shows the different genomes, like the pathways. And as you can see, it's quite a lot there, but you can zoom in and take a look what's going on. And then the Louvain cluster, which shows different clusters. And this one is one cluster I chose, and you get to choose, like, it shows like the words of selected that are of interest, like for example, receptor, apoptosis, cancer, TGF-beta. And then similarity score set shows the Venn diagram of the different like GNPAs and what the field of interest. And then last but not least, the gene expression view. So this shows the like the different like classifications and shows like which thing is being it shows the different expressions according to non-response, response, and weak response. And this one so the conclusions of Pager web app is a powerful tool in looking at different molecular mechanisms, and then Pager web app could be applicable for precision medicine and drug development. And then these are some of the published works, like we've been, And if you don't mind, I'll like to demonstrate the pager web app real quick. So everyone can see my screen.
Yeah, 5 minutes left.
Yeah. Okay, so this is the overview of this is the pager web app, and you start with the facts. Like it says here a little bit of information. What is page and what is a pager web app? And here it shows the R and M types and shows the differences like co-membership gene type versus regulatory. And then it shows here how to cite us, and then of course, and then here it shows the like the Dr. Chen's lab and members, and load the data set. So example we like to use is sclerosis, although you can go click here and use upload data table, so you can just browse and the MS. And then proceed, so you click proceed there. And then it shows the volcano plot of differentially expressive things. So DN is down and then red is up. So it shows this beautiful, and you can adjust this accordingly. This is the default, but if you can adjust according to your needs. And here it shows the gene symbols here, the log change and significance. So you can play around with that, and then you can proceed. And then here it shows the pager. So it shows the enrich and pager and shows the different pathways here. And then the different reactomes, and then the enrich. And then the page-to-page network, all you have to do is press a run network, and it shows you all the similarity pathways and shows the WN10 and like a lot of stuff. So you can just zoom in here, for example, and it shows you this different types of pathways, how they interconnected. And then you show that it shows a little vein cluster, so you can just go select cluster 1, 2, and then 3, and it shows the different classification. Of course, there's gene expression. This you have to upload different matrix, and one here using melanoma since we have the differences in the treatment options. And see, upload complete. And here it shows this in the graph here, and then you have the page ID. So according to page ID, you get to see the response curves according to the gene, and here it shows a gene regulatory network. And then last but not least, can check the comparison of the page wrap versus other. So here it shows the Venn diagram, and you can check different, like for unique terms for pager versus enrich R, and then for web gestalt, and it shows that it's different terms. So,
You have 2 minutes.
Yep. So does anyone have any questions or?
One question, Chandra
Sure.
here, Chandra Brahman.
Sure. Yeah.
Raman, nice talk. So when the database you're inputting for sequences, etc., is it user input, or do you or you can use a combination of user input as well as public databases? I mean, it's a naive question from my part as a person who does.
Sure. It's actually a good question, and because with the page wrap, like originally there was like databases really collected, and actually it's from different databases. Like one database is actually from UAB, but then you can also collect some of your own data, but like if it's made presented in a certain way, like organized by not only the gene, like the P type, and but it's a very useful tool, and we're able to find some pretty interesting discoveries.
I have maybe a question about
Sure.
also. I think it is very interesting. You have found the top pathway which is affected, which is circadian rhythm in the multiple sclerosis, and we can, with Chandra, we can confirm the melatonin is a major regulator of circadian rhythm, and melatonin, it has been described quite recently, has a beneficial effect in the multiple sclerosis. So this online bioinformatic analysis has the reality, in fact, with this what is in the clinic, and this is beautiful because you probably didn't know this, but
All right, thank you for the talk and for the questions. Unfortunately, we have to move on. We're at time now, so time to move on to the next speaker. And next speaker, oops, that's my alarm there. The next speaker is Emma Graffis, sorry, I don't know how it's pronounced. Graffis, okay, Graduate School of Biomedical Science, Division of Infectious Diseases at UAB. The title, Studying Medically Relevant Bacterial Propagation Through Prospective Multimodal Analysis of Clinical Isolates.
Can you see my screen?
Okay. Yep, I can see it.
Thank you. Okay. So multi-drug resistance is an emergent healthcare threat worldwide. Non-commensal, non-commensal gram-negative pathogens have a wide variety of genotypes and phenotypes, but there's only a few which are associated with human disease. And carbapenem-resistant Acinetobacter baumannii has recently been declared as an urgent threat according to the CDC, and the WHO has listed it as a top priority for new antimicrobial development. And there is much left to be known about the propagation, treatment, and control of multi-drug resistant organisms, including carbapenem-resistant Acinetobacter, and previous studies have been somewhat limited. In the past, previous research has focused on phylogeny studies, but now that whole genome sequencing is cheaper and more available, it is a great way to increase resolution of molecular epidemiology. And when combining whole genome sequencing of clinical isolates and robust clinical and isolate metadata, this brings preclinical and clinical research much closer together. And this also allows for research of more clinically relevant determinants of resistance. So whole genome sequencing of modern day clinical isolates is very important to inform medical decision-making within an institution. And with this in mind, clinical Acinetobacter populations in the Deep South have not really been characterized. Oh, sorry, okay. To address this knowledge gap, our aim for this project was to develop a multimodal pipeline to collect phenotype and genotype clinical isolates in order to characterize and investigate the molecular epidemiology of clinical relevant Acinetobacter and other multi-drug resistant organisms at UAB. So this is an overview of what the general pipeline would look like. And first, you want to determine what question you want to answer with the pipeline and set your hypothesis. And so we want to emphasize that we spend a lot of time examining and modifying our pipeline to address these questions. And once we determined our pipeline, we established a relationship with the clinical microbiology lab here at UAB so that we can identify and obtain isolates. And we collected the metadata of each isolate, including associated patient data. And then we perform next generation sequencing and we can perform our analysis. So here at UAB, we have a supercomputer which allows us to quickly process and analyze raw reads. And we have used Anaconda 3 to develop Conda environments for specific tasks. And after annotation, we can assess the core and accessory genomes to answer the questions we defined at the beginning of our pipeline. Prior to coming to UAB, my PI developed a pipeline for assessing Acinetobacter in St. Louis. And the results from this study showed that there was a novel lineage of carbapenem-resistant Acinetobacter species, which was ST499. And he was able to determine that this isolate emerged after the disappearance of other globally prevalent crab lineages. He was also able to determine this by using retrospective studies to define biotypes, and ST499 belongs to biotype 5, which is in blue. And also this lineage was not driven by antibiotic resistance. So while ST499 is carbapenem resistant. It is missing genes related to antibiotic resistance, and that is unlike other crab lineages. Additionally, with whole genome sequencing, he used multiple criteria for identification of ST499 clonal clusters. He used phylogenetics and metadata to determine that indicated that there was expansion of each subclone outside of the hospital system. And the results also showed that there was likely propagation of subclones within nursing facilities and wound clinics. So I'm sure you want to know what's going on here at UAB, and we started collecting isolates in November 2021, and we collected so far over 55 Acinetobacter isolates, and about 30% of them are crab isolates. So we sent our first batch of isolates for whole genome sequencing very recently, and hopefully we'll get that back very soon. We've also identified that there's an ST499-like lineage here at UAB, and we determined that molecularly. And there have been wide phenotype variation among our clinical isolates, and even within the same isolates. And this indicates that there's possible phase variation, which we want to look into more in the future.
5 minutes.
Okay, thank you. And also, Acinetobacter baumannii is characteristically chloramphenicol resistant, but a portion of crab isolates have appeared to lose its resistance. And we have seen multiple isolates like that here at UAB. So in the future, we plan to continue collecting Acinetobacter for another year, and we hope to collect about 200 isolates, with 60 or so being crab isolates. And we will do whole genome sequencing of all those and complete comparative genomic analysis to assess factors related to propagation of crab subpopulations and also chloramphenicol susceptibility. And we hope to adapt these results into preclinical research here at UAB, and also hopefully other institutions will start using it as well. And finally, we will hope to apply and adapt our pipeline to other multi-drug resistant organisms here at UAB and hopefully elsewhere as well. And with that, here are my references, and I want to thank my lab, the Calyx lab, and also the clinical microbiology lab and other labs which have helped us collect these isolates here at UAB. And with that, I'll take any questions.
Okay, hearing none, we're going to move on because we're a bit behind. So we have one more flash talk in this session, and that will be Tan Nguyen, and he will be talking about, he's a postdoctoral fellow in the Informatics Institute UAB. He'll be talking about autocoder finds novel cardiomyocyte clusters contributing to cardiac regeneration from single cell data.
Hi, good afternoon, everyone. I am like Amy said, I am a postdoc, do an appointment at BME department and Informatics Institute at UAB. Thank you for bringing me here, and I also apologize if I prepare the slide in rush or anyone might disrupt me from behind. This is a very critical time, and I have teaching duty and many other duties. Next week is UAB final week, so yeah, if someone interrupt, they need me more than that. So not to waste time. Excuse me. The background. So I try to say, the key message is heart cell, especially cardiomyocyte, is very different from other biological or living system we usually thought of, like, for example, cancer. In cancer, the cause of cancer is a cell growth proliferate, but the cause of, main issue of the heart is they don't grow, they don't proliferate. So, if we have an injury might wipe out a very large number of heart muscle cells or cardiomyocyte, and there is no way, almost no way, the body can make up for that loss. And unless the injury happened when the animals, we tried in pig and in mouse, if the injury was within 7 days afterward, then the animal can have a chance to fully recover. If not, then there is no hope. And then the strategy, and then there's a little more thing about the heart. You know, the heart may respond to injury by increasing the size of the cell or form a scar. Those are very normal in other injury, but these response are not not good in the heart because those response doesn't make the heart beating. If the heart doesn't beat, then we don't need that. So these are the summary of the injury model we try in BME. So to induce a heart injury, we can either choose to cut a bottom tip of the animal heart. That is called apical resection, right there on the left side. Or we can simply block the blood flow in the heart vessels. Then that after about 5 minutes, then there's a large amount of cell, you know, they die simply like a blood supply. So these are the two injury model we try in our lab. And then the third one, I have never had an experience on that. But basically the cool finding we got, first of all, David Chow is a good friend of mine. What he did was he cut these, you know, the pig heart, you know, apical resection when the pig is 1 day old, and we know that the pig will recover. 1 month later, we know he knows that the pig recover, and he induced the second injury, the infection, and then the pig, very surprisingly, they recover by regeneration, confirmed by, you know, proliferative checking. Proliferative cardiomyocyte checking. So complete regeneration and no scar. And this is among the first time in the field that we find a way for the heart to naturally regenerate after they pass 7 days time point. And of course, it's not totally natural. Natural regeneration, because there is a little trick when we cut the heart on day 1. And then including me and Nakada, using Nakada, we follow up with single nuclei sequencing. We wrote a paper and we published that very recently. And so this is about, you know, what we reported in the paper. So the idea is we take these regenerative, you know, double injuries heart, they regenerate. We took those hearts, send them to single nuclei sequencing. We try to find cardiomyocyte subpopulation that hopefully contribute to the regenerations. And so the landscape says we identify 6 subsets of cardiomyocyte or heart muscle cell, and we focus on this thing called CM5 over there. This subpopulation overexpress PKM, which we know to be a regulator of cell cycle. And also very interestingly, later on, you know, we can see it right there. The tryptophan T3 is the marker of smooth muscle cell. Not that. The skeletal muscle cell, and we know in the field that skeletal muscle cell and cardiac muscle cell usually exclusive. There is usually no skeletal muscle cell in the heart. So this is interesting, and we are trying to investigate. So this is one of the remaining questions from our past study. It looks like it contributes to proliferation, but we don't know how. So that is why we move up with my research later on, and a few more things about background. So I think this popular for most of the people doing single cell. We need to normalize the data. We need to cluster the data to find cell subpopulation, and then we try to annotate those cell population by pathway analysis.
You have about just under 4 minutes.
Yeah, the challenge in those is, you know, so by myself, not because they are failed by techniques, but by finding the thing we don't want. Because the cell in the heart are complicated, we might find cell that does not proliferate, but doing something else. So objective, certainly find population that contribute to the regeneration. So the method, in summary, I try to do on autoencoder. So basically, this is a model, computing model. It's a deep learning based network. And then the idea is we build up the architectures, and then we take the middle layer of the architectures and then use the data at the middle layer for clustering. And what my claim, there's no method good for everything. So better to check before we decided to use any pipeline in any data set. And then we also use the sparse model, basically to classify two groups of cells based on a specific cell cycle process of signaling pathway. If this pathway can separate those two cell subgroups, then it is an indication of enrichment. The results is what we just sent to a journal, and it's under review. So I reanalyzed and I found a unique subpopulation called CM1 in a regenerative heart impact reception P28. Like I mentioned at the beginning, they regenerate. So
you have 2 minutes.
Exclusive. And we also found exclusive cluster for fetal heart, which should grow and proliferate. And then we analyze cell cycle for those subcluster. We don't, this doesn't tell, CM1 is proliferating, but it has the regulators for cardiac muscle proliferation as much, as high as the fetal. And then these are the 4 unique markers for the CM1. So all of them, TBX, well studied in our lab. GRK5 are normal, is normal. ERBB4 is also, a promising one. And then we try to do the downstream genes of those 4 markers and found many signaling pathway and process, promoting proliferations.
You have 1 minute.
Conclusions. Yeah, that is the last slide. So we found a new subpopulation of myocyte that contribute to proliferation, and we want to focus on GRK5 marker. The other marker are being studied also in our lab, so I'm quite happy with the result when it is useful for further validation. So acknowledgement, I will thank our event chair and then PI, Dr. Jay Zhang. Juji is a good friend of mine sitting just next to me. Zhang Zhou is a person who's have a lot of idea. TBX, she is an expert on the progenitor cell, and Jake Chen from Informatics Institute. So this is the end of my talk. I hope I have time to answer some questions.
Unfortunately, we have to move on. So if there are any questions, please enter them in the chat. Thanks very much for the great talk.